Okay, I think we'll begin. I'd like to thank everybody for joining us today. And I'd also like to thank Dr. Mark Williamson for agreeing to present as part of our didactic training series for Transcend. With that, I'd like to introduce Dr. Williamson. Mark Williamson is an assistant research professor at the University of North Dakota School of Medicine and Health Sciences for the Population Health Department. He serves as a statistician and coordinator for Transcend's Research Design, Compliance, and Data Management Core. He has a broad scientific background that includes physics, analytical chemistry, biomedical research, statistics, ecology, genomics, and epidemiology. His current research is exploring radon exposure as a risk factor for various cancers and developing experimental radon projects, which include small radon chambers for remedial testing, cell culture exposures to radon, and an experimental radon test chamber for animal experiments. With that, I'll turn it over to Dr. Williamson.
All right, thank you so much. This is my talk for the Didactic Lecture Series, Hypothesis Generation for Research. So, last month, I lay awake, thinking, sweating a little bit, thinking, wondering if the package that I ordered was waiting for me in the dimly lit second floor medical school post office. The package I was waiting for was biochar. So what the heck is biochar and why did I care so much about it? Well, it turns out biochar is a form of carbon. It can be added to the soil to improve soil health, but it's also, because it's a form of carbon, I had a very specific hypothesis. I thought that if I added biochar to a chamber filled with radon, it would absorb some radon. I came to this hypothesis because I've generated a lot of ideas, filtered them out, and am ready to test the most promising ones. And in fact, it turns out that activated carbon, activated charcoal, another form of carbon, does totally absorb radon. So there's a very short leap between that and biochar, another form of carbon, to test this. So that's an example of coming up with a bunch of ideas, finding ones that actually work that we can test the hypothesis on, and moving forward with that. So that's kind of what my talk is today. So let's start sort of at a high level here and talk about what we mean by the term hypothesis, and then we'll get into sort of the concrete ideas, how to generate a lot of research ideas to be able to turn into these hypotheses, and how we can filter those into testable hypotheses. So what is a hypothesis? You could start with looking at the etymology, so what the word means. Comes from hypo, which means under, and thesis, proposition. So you could think of a proposition that is under consideration. Now, if you look at a definition, look up something like the Merriam-Webster. Their second definition is very helpful. It's a tentative assumption made in order to draw out and test its logical or empirical consequences. Now, that definition is fine as it goes, but it's a little clunky for my purposes, so I'm going to suggest a second MW definition, the Mark Williamson definition, which is a hypothesis is a testable answer to a useful or interesting question. And the words I use to bring up this definition will matter, and we'll go through it later. Now, hypothesis is certainly part of the scientific method. If you look up images of scientific methods, you'll get 1,001 pictures like this. It's usually circular, that is iterative, and a hypothesis falls somewhere usually in the middle. The stuff that comes after is pretty straightforward. You run experiments, you analyze that, you have conclusions. But some of the stuff beforehand, how you actually get to the hypothesis, is less clear, a little bit more vague. And I find that there's really not a lot of formal training on hypotheses in formal education. A lot of scientific education, while very good, focuses more on what I call facts and procedures. You go to Biology 101 and you learn a lot of facts about biology. parts of the body, the Krebs cycle, taxonomic classifications. And then you go to something like cell biology lab, where you learn procedures, how to run a Western blot, how to use a microscope, things like that. And also, since this didactic lecture series is about clinical and translational research, if you're a clinician, you're a doctor coming in from the medical field and want to do research, your training probably also was a lot of facts and procedures. Here are the diseases, here are the symptoms, and here's the procedures, how to treat it. And so some of that training on how to actually come up with hypotheses, come up with ideas, is somewhat lacking. Now I want to move on to, again, going back to my definition of a hypothesis, a testable answer to a useful or interesting question. Now, testable is important because that's what makes it science. We can actually test it against reality and see whether we're right or not. And then the next two words, useful and interesting, are very important. Thinking about sort of science on a more philosophical level. I'm really interested in the philosophy of science. And so I like to think about that. And because this is my talk, you're going to get roped into that for the next couple of minutes here. So just bear with me. So we can think of science, so philosophy in the wide sense, as being subdivided into other types of sciences, other types of knowledge. Now, we're going to bracket off the methodological sciences and not deal with that here, but talk about maybe what's a little bit more familiar, the pure sciences, or what's sometimes known as the basic sciences, and the applied sciences, the practical sciences. And the reason I use the terms useful and interesting is because they often dictate what kind of question, what kind of science we're doing. Now, a useful question is almost always an applied science. So that's something like engineering or like medicine, biomedical research, clinical and translational research. Your questions usually want to have some sort of practical output. Whereas something that's simply an interesting question is maybe more fit for a pure science, a basic science, truth as truth. Not to say that you can't have intersections of both useful or interesting. That's probably the best question. But it's helpful to think of what kind of questions we're even answering given our goals. Do we doing applied science or are we doing pure science? So now let's talk about this. Okay, we have a definition for hypothesis. We can kind of think of broad levels of questions we can ask, but how do we actually get there? So strangely enough, developing a hypothesis actually has two sets that we maybe don't think about, two areas that we don't probably think about so much in science. There's a creative area and actually a logical area. We tend to think of science as sort of very cerebral, purely logical, but there's often a very creative aspect that needs to go into that. And I think hypothesis generation is one of those. And in fact, a lot of very important and often challenging areas require both a creative aspect and a logical aspect. Like, for example, if you're going to write a novel, you maybe brainstorm and you have all these creative story ideas, but then eventually you need to be able to beat those into shape and create some sort of logical structure like a story outline. So too with research. To really get great hypotheses to test, you're going to need to come up with a lot of research ideas, which is a much more creative process. And then only after you have those ideas, you can sort of whittle them down, beat them into shape, you can come up with some promising candidates that you can turn into a bona fide research plan. So that's what we're going to try to go through today. Talk about the creative side of generating ideas, and then the logical side of sort of filtering those ideas down. So that's where we're going with the next part of this talk. We're going to talk about how to generate a lot of research ideas. And then with those ideas, we're going to talk about how to filter them down to ones we can use in a research plan. So that's the question here. How to have a lot of ideas. I have 3 suggestions that might help you on this. And then the fourth one is sort of bringing it all together and capturing those ideas. And really, the first three, reading a lot of papers, visiting a lot of places, and talking to a lot of people, all have the same outcome in mind. It's to get the ideas in your brain that are sort of bouncing around, to get your brain in contact with as many other ideas out there and bounce as many other ideas off yourself as possible to come up with new ideas, to spark insights, to help generate these ideas you can put into what I call the off the walls folder at the end. So let's move along here. First suggestion, read a lot of papers. So scientific papers, that's what I'm talking about here. So the impetus, the reason to kind of do this is, well, if you want to solve problems, do research in an area, you probably need to know the field. Say you're interested in thyroid cancer, you probably need to know a little bit about thyroid cancer, what other people have done, how it works, knowing the field. And most importantly, especially when it comes to solving problems, you need to know the gaps. What don't we know about thyroid cancer? What are the mysteries? What are the contradictions? Those are going to be very fruitful areas to come up with testable hypotheses. Because if we don't know something about thyroid cancer, well, that's a great place to test. In addition, it's helpful to read a lot of papers, and not just in maybe your narrow field, but as widely as you could possibly stand, because that'll give cross-discipline fertilization. Other disciplines have other problems, have other solutions, gives you much different ideas to bounce your ideas off. It widens your scope. And then finally, reading research papers from scientific journals, it gives you experience with what I call reifying ideas. So to reify is to make real. So that's a scientific paper in a nutshell. Here's an idea we wanted to test. Here's how we tested it, and here are the results. And this is especially helpful if you're just getting into research or you're getting into a new field of like how people actually do things. Sometimes the method sections in a paper is the most interesting thing a paper contains. So that's some of the reasons to read a lot of papers. And to help you with that, I've been thinking through some tools to maybe help you on this. So the one that I start with, and I think is sort of the baseline one that's really helpful to read interesting and helpful papers, is a site called Science Daily. It gives summaries and links to papers on a daily basis. I really like to look through this weekly and just find the most interesting papers to sort of mark them down. You can also, if you're just interested in a subfield, chemistry, medicine, whatever, you can filter by that. So you can just look at a subset if you want to too, or you can look at sort of the whole gambit of science. And so that's a really helpful way to come into contact with a lot of interesting papers. The next tool, sort of building up on that, is some code I've created. If any of you are a little bit more computer, Python minded, on GitHub, I have some, I would say maybe mediocre Python code. And I'm going to throw that in the comment here. And then this will be for the recorded video. This should be in the description. Because I would have loved when I was younger to have a source that I could just say, give me a random scientific paper to read. So I don't have to scroll through things, just prompt me with something interesting. Now, I'm sure at this point you could use AI to give you a random scientific paper, but if you want to kind of figure out how to do it yourself, here's some code. And this takes from Science Daily to read a random scientific paper a day. And of course, your mileage may vary on this. This might not be helpful at all, but maybe you can take the idea of this of finding a random scientific paper that's maybe outside of your normal comfort zone to help generate ideas. And then finally, the third tool here is I actually have a podcast called Science Research Weekly, where I go over what I think are some of the most interesting papers, tools, and even grants of the week. Sorry, I put that in the chat here. And it's about 10 minutes if you want to use it on your weekly commute home, I go over what I find the most interesting papers are, and that might be a helpful and low level way to get more experience with potentially papers of interest. So take all these with a grain of salt. I would say if you want to start, I would start with Science Daily. And again, because the more papers you read, the more ideas you'll get in contact with that you can bounce off your ideas to generate new ideas. Second tip to generate a lot of ideas is to visit a lot of places. The impetus for this is, well, if you go places, go to symposiums, you go to conferences, etc., that's going to be where the opportunities are. This doesn't have to be, in far-flung Iceland. It doesn't have to be across the country. It could be just at your local university or local institution. There's often a lot of great, free, interesting opportunities if you look for them. And it doesn't even have to be in person. Well, in person is often the best, so the brick and mortar. There's a lot of online opportunities now, online webinars, symposiums that you can get to despite maybe having a busy schedule. You can pop into a couple of sessions online. It's really only limited by your ability to pay attention and to look for these opportunities. Reasons why you should visit places. That's where the people and ideas are. People are endlessly fascinating, and if you give them a chance to share, they will inevitably surprise you. And finally, visiting a lot of places helps shift your attention and your aim, get you out of your lab, get you out of your comfort zone, because you only can sort of know or see opportunities if you're looking for them. You don't know what you don't see. So if you deliberately get yourself out to different areas, different places, different conferences, that'll help shift your attention. And some tools for this, as I kind of noticed, you can go to anywhere, but local conferences are a great idea. Also local poster sessions, symposiums. One of my favorite ones at the University of North Dakota, there's this thing called the 3-minute thesis competition. It's where graduate students present their work in 3 minutes or less. It's a competition. It's one of the most succinct, exciting, and bite-sized way to hear about other people's research. And so every time I go there, I'm actually alumni of that. I competed that as a grad student, but I like to go see it as a faculty member now. It's the greatest bang for my buck when it comes to learning about new ideas. And so you can find your own 3-minute thesis equivalent. Of course, there's a lot of great virtual workshops. For example, I went to NDSU's Center for Computationally Assisted Science and Technology training sessions on high performance computing. That was free. I could even watch it sort of after the fact. So that was a very low-lying fruit when it came to learning about new ideas, learning about new procedures. And then finally, this is my personal favorite. Your mileage may vary. I love going to libraries and going through the books and just pulling out random books and finding interesting things to read. Some great examples at the local UND Chesterford Library. I found this one of the best encyclopedias on animal life, which I cannot pronounce. It's, I think, Grisomix. It's multiple encyclopedia. Really the best resource I've ever found of a high-level view of animal life and its diversity. At my local public library in Grand Forks, I found this trilogy called The Wizard of Ads. It's all about how to advertise, and especially advertise in sort of a magical way, in a way that it doesn't just sound like an ad, that it helps tell stories. And I found that that's very helpful. Finally, I think this was a interlibrary loan, and trying to maybe pull it back to more the clinical translational research. I found a recent book called Blind Spots: When Medicine Gets It Wrong and What It Means for Our Health. Really eye-opening about, despite our best efforts as a society, as as clinicians, as scientists, sometimes we get things wrong. And it's important to recognize that and learn from those blind spots. So then finally, one way to generate research ideas is to talk a lot of people with a lot of people. And this should follow from the first two, because if you're reading a lot of papers, well, papers are written by people. If you go to a lot of places, well, places are where the people are. So this should sort of follow from that. Again, humans are endlessly fascinating. So if you listen to them, talk with them, they'll share their expertise. They'll share their perspectives. And this is a great way to find collaborators. Some tools. Obviously, you can just talk to people, in your office. You can talk to people at conferences. But here are maybe a little bit more off the beaten path pathways to tools for talking with people. The first one is papers. If you're reading interesting research papers, well, guess what? All the authors are listed on these papers, and they usually have a corresponding author with an email that you can actually reach out to people. For example, there's this article about synthetic yeast genomes. I read it actually, I think mentioned on my podcast, and one of the corresponding authors of this paper actually reached out to me and said thank you, and we had a nice little correspondence. So that was a cool connection point. Again, where places, if you go places, you'll run into people. One of my favorite things at UND is there's this thing called the Space Ag Conference, agriculture. I love going there. It's very different from my field, so it gives me a lot of ideas. And in fact, I met someone there who has a CEO of a self-funded company, Gravity Grains, who I actually formed a collaboration with. And he's actually the one who sent me that biochar that I can now do research with, even on things like radon. And then finally, again, this is a newer thing that I kind of discovered that I'm playing around with. And, given your own temperament, your mileage may vary, is deliberately interviewing people. I went through this program called I-Cores. It's part of the National Science Foundation. There's this Great Plains I-Core session. It's really about, I-Core is about helping scientists who maybe have developed a product to be able to transfer that into, say, commercialization, turn that into like a business. And so I had some ideas, not a business yet, not even a product yet, about radon reduction. So I went through that, and a huge thing we did actually was interview people and learn about what they would want in a product, because it turns out the number one reason a business fails is because they build a product that no one wants. And so I interviewed a lot of people about radon reduction, not landlords, homeowners, renters. I talked to public health specialists. I even talked to radon mitigation experts, and I actually talked with someone, a radon mitigation expert from Fargo. This is fascinating and unprompted. I mean, he shared all sorts of great radon information, including some potential radon research that I could do that I didn't think about, that I'm actually going to follow up with. So by talking with someone, by interviewing them, I was able to, without trying to sort of tease it out myself, freely shared with that, from that endlessly fascinating person, I got research ideas. Okay, so we've talked about in generating a lot of ideas, to talk to a lot of people, to go to a lot of places and read a lot of papers. What then? Well, the fourth part is to sort of wrap it all up, is to actually capture those ideas so we have them for later. So that I use what I call off the walls folder. You think of this as out of the box, throwing things against a wall. It's really just a folder on your computer or whatnot with some quick text documents that just document your ideas. When you have ideas, that you generate, you put them down. You can add whatever notes, or you can expand on it. You can add references and really just save and review that. So now this off the walls folder has a storehouse of all these ideas. And remember, this is a creative process. The idea with this is to put down all sorts of ideas, whether they're good or bad, whether they're feasible or not. It's to build a storehouse of creative ideas. the next step is we'll actually filter them down. So don't worry about filter them first, just get them all out. So I have up on the screen here a couple examples. I think I started this back in 2022, so I have some initial ones that I bolded point in a Word document. My more recent ones, I just have a text document and I just go through every month and I jot down some ideas. They can be fairly straightforward, like referencing a paper, like radiocarbon batteries for medicine in space. It can be sort of totally off the walls and probably totally unfeasible, like a rotating air filter to collect environmental DNA attached to long haul trucks. The idea isn't that these are ideas that you can totally do. It's to, again, capture the ideas you've come up with so you can filter them later. So with that, let's talk about that next step. We've gone from the creative to generate a lot of ideas to now more the logical. How do we filter those ideas into testable hypotheses? So I have a couple tips here. Again, some of these things is what I've been developing myself, either consciously or unconsciously, and trying to get better at myself. So take all of these as far as you will with yourself, see if they work. They might not work for you. Sometimes they don't even work for me. So think of this as yet another idea that you can filter and test. So we have our off the walls folder full of a bunch of ideas, some good, some bad. Really, the amount of good ideas you have is contingent upon the number of ideas you have. So the more ideas you have, the more you have to filter through and find the really good ones. So to start by filtering, I have what I've put together an acronym called SWEET. This is more of the high level, sort of almost intuitive or sort of gut checking to help take a lot of ideas and filter more promising ones. So the SWEET stands for spellbinding, useful, important, timely, and executable. So first up, spellbinding. It really should be science that grips you, research problems that grips you, because science, doing real good science, it's really hard. So you really should have problems that grip you, problems that motivate you, despite all the difficulties, despite all the obstacles. I think that's one of the most important things. Work on problems, if you can, if at all possible, work on problems that really grip you, that are spellbinding for you. Next up, especially on the more clinical, translational, the applied science track, you probably want a question that's useful, that you can, have some sort of impact, reduce cancer risk rates or something like that. A similar idea is it should be important. Probably should be a trivial question. Now, useful and important, they kind of overlap. I think of useful as more of like a yes or no switch, like either a research question or research idea is useful or not, and then important is how useful it is or not. So it should probably be useful, yes, and then from there maybe you have a couple ideas. Both are useful, but one might be more important than the other. Maybe one would reduce risk of some sort of disease by 5%. Maybe one has the possibility for like 10 or 15%. That's a little bit more important. Or maybe it's easier to implement. Another sort of gut level thing to think about to filter the first round of filtering is timely. Is this the right time for it? Could you do it right now? Do you have the space? Maybe it's a great idea, but it's not a good time for you. Maybe the funding cycle isn't a good time. Maybe next year there'd be a funding opportunity you could shoot for. So sometimes timing can be everything. And finally, it should be executable. Are you personally able to do it? Maybe you don't have the money, equipment, or location yet. It could be the greatest idea in the world, but if you can't execute on it, it'll remain an idea. So you can use this to sort of filter down maybe a whole folder full of ideas into 10, 15 more promising ideas. Try to find that sweet spot, as it were. Once you've kind of gone through maybe the SWEET method or something similar to filter a bunch of ideas into a few more promising ideas, it might be helpful to go through the next step, the next sort of filtering, what I call the white paper method. So you might have great ideas, but they might be too fuzzy or vague. Like there's not enough detail to know whether or not they're really practical. So that's where doing something like a white paper is. would come in. I consider paper something like an informal proposal. It's not like a grant proposal, which is here's a really detailed, intense thing. Please give me money. It's almost even more for yourself. You maybe not show this to anyone, or maybe you'd show it to your boss or higher up or something like that. Really, it's a way to flesh out an idea and see what it would take to put into action. It should be fairly short. It should have broad details of like what you want to do, but also should have some clarifying details of like how you would actually do it. Some parts of it, should probably bring up why it's important, either to yourself or more broadly. Going a little bit into the experimental design. How are you going to test this? Are you going to do a chart review? Are you going to do a clinical trial? Are you going to do a survey? Like how you actually gather the data you need to answer the question. What are your expected results? And of course, to make this a real thing, how are you going to fund this? Is it free? Do you have institutional funding? Is there a grant opportunity? What kind of grant opportunity would you need to do this? An example, sort of from my own work that kind of fits into this white paper, maybe a little bit more intense than white paper, but on the similar track, is several years ago in 2021, the Dakota, the grant before the Transcend grant that I'm part of now, had a call for equipment funding, and I wrote a white paper essentially to get funding for equipment for a radon chamber for small animals. And I was able to lay it out of like, this is what it would actually take. You can't buy a radon chamber, you have to build it. So it helped me figure out how to actually build it, and actually was funded. And so I have the supplies. Now I need to find a place to actually put it together. I think this is an important point from the second didactic lecture series that happened last month, that Dr. Soo-Jung Kim really brought up the power of small wins to get where you want to go. So a white paper, a small research project, it might be small. You might not have a lot of funding for it, but if you can get a win there, you can make a small idea real. That can get you to the next step here. So never underestimate the power of small wins, of making a small idea as real as you can. Okay, so got a lot of ideas, maybe filtered them down a little bit, maybe wrote a couple white papers or something even less formal, just jot out what it would take to make these ideas real. Eventually, you're going to have to make a selection. You're going to have to say, of these ideas, these are the ones I actually want to move forward with. I want to actually try to do. And so I was thinking with myself here, like, what are some of the go, no-go questions that you really need to answer to make a project work, to make an idea into a hypothesis that you'll actually test. So I came up with these criteria: a feasibility plan, a certainty of funding, a could do versus would do analysis, and internal versus external support. So sort of asking yourself these questions. If it's a yes, great. If it's a no, it might not be a thing you can move forward with at this point. So I just, as an example, I sort of pulled 4 random ideas I had from one of my lists of off-the-wall folder that I can kind of go through here. So the first one here is, these are these essentially small business or like tech transfer programs. Again, coming out from the I-Corps, having researchers who have products, getting them to market. So there's sort of this extension program of what I did here. And I thought, well, okay, is this feasible? Well, no. I don't have a product yet. Maybe I will in the future, but I don't have a product to sell. I don't have funding for it. While I could do it, I don't think I would do it at this point. And while I do have support, maybe from I-Corps, this really isn't an idea that I could really select at this point. Another idea, I was reading about these things called obelisks. They're a type of viroid, which is like smaller than a virus, which is really fascinating. And I would love to explore more about that. So thinking through these questions, totally feasible. I don't have any funding for it, though. I think I could do it, and I would do it, but I don't have any support. It's totally outside of my field. So as cool as this is, it's probably not a good selection. The next two come from some papers I've recently read. One was about radiocarbon batteries, and I thought, oh, like you could co-op that for medicine in space. I think it would be totally feasible. Fortunately, I don't have funding in that area, and I don't even know if I could do that at this point. Though I totally would if I could. And I don't really have support. So while this is promising, it's not the time. Finally, there's this paper on do-it-yourself filters for indoor air health, because I work on radon, which is a health hazard. I thought, well, maybe there was a way to incorporate radon reduction into the air filtering. Is this feasible? Yeah, I think so. It's pretty low cost, pretty straightforward. I mean, some of the radon things need to be solved. That's why it makes it an interesting problem. And I totally could, I'm sure I could get some funding for this. It's not very expensive. I have a lot of institutional support for radon. I could do this. I totally would do this. And again, I do have institutional support. My department does a lot of radon research. So of all these things here, that the do-it-yourself filters for indoor air health is an idea that is ready for prime time. It's ready for selecting to actually turn into a hypothesis to test. All right, so we talked about that filtering, going for a lot of ideas, some good, some bad, filtering out the better ideas, thinking through those ideas to flesh them out, and finally making some hard selections. The final step here is more about reviewing. If you have your off the walls folder, you have that forever, and you're hopefully continually adding to it. And so it's helpful to regularly, review, maybe every year or maybe every couple months, of just going through your ideas and seeing what you have there. Because a lot of ideas, they might be great ideas, but they're not fleshed out. Maybe you need more information. Maybe when you go back to it, you have more information to flesh it out. Or maybe it wasn't a good time. You didn't have the funding for it, but now you do. The image I like to think of is, it's good to have a lot of like sort of pots on the back burner, sort of bubbling. You're not working with them right now, but you kind of have them simmering. And when the right circumstances come up, you can just pull them up to the front and be ready to go. And I think that's a really helpful mindset to have, is have a lot of interesting ideas on deck for when their time comes. All right. With that here, I think we're doing pretty good here. So hopefully that was helpful. But I thought, while I have you here, why don't we start putting these ideas into practice with a quick exercise. So what I'm going to do here is I have some more Python code. I think I put it into the chat at some point, that essentially I can take and just generate some random subfields of science as a prompt. So what I'm going to do is I'm going to have some prompts for health research in a random field. And then what we're going to do is, once we've got that prompt, I'm going to give everyone watching here 5 minutes to just brainstorm ideas about like interesting questions that you could answer that are based on those fields. And then we can even get our off the walls folder started and talk about it. So what I'm going to try to do here, stick with me here. Okay, does everyone see, Kent, do you see a screen with some text boxes open of Python?
Yes.
Okay. All right, so I have some more code. I'm going to collide some ideas here. I want 5. And then we're going to have health research and a bunch of other random subfields. Okay, so we have 5 here. Health research and atmospheric science, health research and dynamical systems, health research and vector space, health research and computational number theory, and health research and neuroscience. Why don't we take the first one, health research and atmospheric science. So what I'm going to do here is go back to my slides. And so our random field is going to be atmospheric science. Is, let me just say at most, there we go. That's close enough. Atmospheric sciences. So what I would like you to do for the next 5 minutes on a pad and pen or just on your computer in a text file. I want you to brainstorm ideas that intersect health research and atmospheric sciences, and come up with as many ideas as you can in 5 minutes, and then we'll come back here and talk about it. So I'll get started here. I have a little timer. Once all these green bars go out, our time will be up here. So enjoy. All right, that is 5 minutes. All right, at this point, hopefully you've jotted down a couple ideas, maybe just one idea, maybe I think I got like up to 1, 2, 3, 4, 5, 6, 6 or 7. It was uh a little easier for me because atmospheric science, I think radon and health research, and that was already in my field. But I was trying to think a little bit outside the box. But hopefully you were able to generate some ideas. And now you could add them to your very own off the walls folder. You can, find an empty space on your desktop, right click, add new folder, call it whatever you want, call it off the walls, call it whatever, and then save a text document with these ideas in there. With that, does anyone want to share any of the ideas they came up with that intersect health research and atmospheric science? You could type them in the chats, the chat. I know one of the most interesting one I thought about was especially trying to get outside of just like radon and human health, sort of basically, was looking at ionizing radiation. So there's more ionizing radiation exposure in the high atmosphere, like pilots. It's like looking at, well, does ionizing radiation change the human microbiome, and look at maybe like pilots or stewardesses or whatnot on that. So I thought that was maybe an interesting question. I don't know, anything anyone else has to share? I mean, if it's a little too crazy, a little off the walls for you. Oh, great question, or great idea in the chat here. How does exposure to pollutants or wildfire smoke affect child health? Very broad, but that's a great start, and you could drill down from there. So great example of health research in atmospheric sciences. All right. Any ideas you had, Kent? Not to put you too much on this spot. Another response. Comparison of health outcomes and air quality. Yep, that's great answer. Wildfires on cognitive function of older adults. I know there's a lot of wildfire smoke the last week. Great example on people's mind of what it could actually work, or what it could affect. So yeah, great examples, everyone. Okay, the effect of climate change on pollen exposure and asthma. There we go. Well, I guess with that, we've come to the point where we can start to close. We can open it up for questions. I think Kent can moderate that. If you can push back on me. Like, a lot of this is me trying to think of helpful ways to generate ideas and whittle them down. If you think my ideas aren't great, we can come up with better ideas together. And of course, want this to be a stepping off point for you. We'd love to hear more if you have ideas about where you want to go with generating ideas. I'm happy to talk more offline here. I have my contact information up on the screen, and we can put it in the chat too. So that, Kent, if you want to moderate.
Sure. We have a question here for you, Mark. Clinicians and researchers are often busy, so they may not have a lot of time to dedicate to generating hypotheses. What would the most important takeaway method from your talk be?
Okay. Yeah, so that's a really good question. I would say then, I think a lot of what all this hinges on is having ideas in the first place and capturing them. So I would say something along the lines of the off the walls folder, of having a place to put ideas when you think about them. Yeah, maybe you don't have much time to read many papers or go many places, but you're still going about your day, and you might have ideas, especially if you practice coming up with ideas. Having a place to keep them, and almost more sort of on a higher level, giving yourself the permission to throw ideas down, whether they're good or bad, I think would be my most important, if you only get one thing out of this, my most important point.
Great, thank you.
And can I add to that, Mark?
Sure.
Yeah, this is Paul Carson, and
Yeah.
I'll be doing next month's didactic, and I'll play off of what Dr. Williamson talked about here, but mine will be a little bit more addressed towards clinicians. And I just would say to the people who are in clinical practice that your own practice is really a wealth of places to explore. You're going through your rounds on a daily basis, your clinic on a daily basis, and you're confronted with all kinds of things that you do on a regular basis, often without, I don't want to say without thinking, but without questioning. And I think the easiest way there is to just being curious about what you're seeing and what you're doing in your practice and asking questions. And that's been a real fruitful thing for myself. Like, why do we do this the way we do this? Is this really making a difference here? Is this best practice, alert that's popping up on my computer all the time really changing the way we practice it? And these are often very practical things that can make a difference in your department, in your hospital, in your health system. And I'll go over some examples of that. But I would say, you're the expert in your own field, and being curious about what you see and what you do, and asking questions about that can be very, very fertile ground for generating questions that you can test with a research study.
Great, thank you. Yeah, that's an absolutely excellent point.
Mark, I have another question for you.
Mhm.
Coming up with great research ideas is all and good, but how can we find funding for our project ideas?
Yeah, that's a big thing. I tried to put that up in some of the filtering processes, because that you can have the greatest idea in the world, but if you can't get funded for it, well, then it's going to stay an idea. Now, I would say, you know, uh, one thing is, if you have this folder and you have these ideas and you kind of again have that review, that'll help you sort of keep in mind of like what ideas you maybe have, so that when there is a research funding opportunity, you can catalyze on it. Now, to actually find research funding, I would say certainly if your institution has alerts. I know at UND and even through Transcend, we'll send out funding alerts, checking things like the NIH, our grants.gov, even like Grant Forward, like just getting into the practice of looking for grant opportunities. I especially especially if you have an idea and it's a small idea, finding ways to small funding opportunities are really a way to get a lot of leverage. A lot of you institutions will have like internal funding, that's maybe a place to start. Um, Again, if you're listening to these didactic training sessions, if you're a part of Transcend, you get our updates. We have yearly grants we put out. And if you have an idea in clinical and translational research, that'd be a great idea for that. Looks like someone in the Sarah in the chat gave a link to UND research funding opportunities. So I would say, yeah, try to get more familiar with where funding comes from, funding opportunities, and just sort of have some ideas ready, so when you find a funding thing that matches it, you can act on it.
Great, thank you.
Kent, this is Steve.
Hi,
Could I
Steve.
ask Mark a quick question?
You bet.
Hey, Mark, so thank you. Very nice talk. And I appreciate it very much, the different ways that you've got people just thinking about kind of innovation and new possibilities of hypotheses. My question is, it's a little bit on a little ways down another dimension, and that is for some of us who have been chasing a particular program of research for a long time, we've kind of narrowed it down into this esoteric, narrow little pool of ideas. And so, for somebody like me who does eating disorder research, let's say I think I've got a risk factor, and let's say it's growing up in a particular kind of family or neighborhood, and that increases the risk of an eating disorder. And then I start to think, what's the mediational mechanism? Is it some self-concept thing? What is it, a reward thing? Is it a brain activation thing? What's the process by which the risk gets enacted? But then I also add a moderator and I say, yeah, but this mediational process is only valid for this group of people and not for that group of people. When I start to go down this rabbit hole that many of us are in, the question is, where do our hypotheses come from? And it's an interesting question. It seems like they come from the work. But I'm wondering, do you have thoughts or guidance on that?
Yeah. Thinking about being, a very specific field. And I think it kind of goes back to Dr. Carson's comments is I think the hypotheses, these questions come from almost like what grip you, I talked about what are sort of spellbinding, but in sort of the negative sense, like what bothers you? It seems like that's probably a real source of research questions, especially if you're very familiar in the field of what do you notice that bothers you, that doesn't seem right, that in some senses annoys you, of like, why is it this way and not that way? Like what could be the issue of that? And if you could find problems that bother you, and you are in a position to solve them, I mean, it's probably hard to think of a better spot to be in when it comes to being a scientist, when it comes to being a clinician, even though in the midst of it is sort of uncomfortable. Finding problems that you can personally solve, I think, is great.
Yeah. Thank you.
We have a couple other resources posted in the chat as well. I'd like to thank everybody for attending today's didactic training, and I'd especially like to thank Dr. Williamson for his presentation. Lots of great resources and excellent ideas. I'd like to also remind everybody to please take a moment to take the survey. The link is found in the chat. Your feedback is greatly appreciated. It helps us improve our didactic training series. With that, I'd like to also wish everybody a great rest of their day, and thanks for attending.